about the differences between smooth and cardiac muscle in terms of their structure and their function. We've talked a lot about skeletal muscle, the sarcomere, the myofibrils, the factors of force generation. We've talked about excitation, contraction, coupling, and how muscle contraction happens in skeletal muscle. And we're going to kind of do the same for smooth muscle as well. So let's start out with a few of the differences between smooth and cardiac muscle. And I'll also be talking about skeletal muscle since we already looked at the structure of that muscle already. So smooth muscle, although it shares some properties with cardiac muscle in that they're both involuntary, we don't make conscious decisions about, smooth muscle contraction, blood vessel contraction, cardiac muscle contraction. Structurally, they're very different, right? Structurally, smooth muscle is nothing like cardiac muscle. It doesn't have the sarcomeres, therefore it doesn't have the striations that we see with cardiac and skeletal muscle. It's found in internal organs, so think about the bladder, think about the uterus, think about your gastrointestinal tract from esophagus all the way through. And we said blood vessels, yep. So the internal organs and blood vessels are where we mainly see smooth muscle. Again, it's under involuntary control. So that's going to be control of the autonomic nervous system, meaning that we don't consciously decide to constrict blood vessels or to, contract the bladder. The cells here are smaller. They're about 1/10 the size of skeletal muscle cells. So they're not those large elongated fibers the way that we saw. And we've got this illustration showing that here. So for skeletal muscle, we had these long elongated fibers. They had many nuclei. And then here in smooth muscle, the cells are much smaller. They're spindle shaped, sometimes referred to as leaf shaped or pinnate shaped. Again, much smaller, and then they have their own nuclei. They also have actin and myosin, but the actin and myosin not only look different, but they're configured differently. They interact with each other differently as well. There are no sarcomeres again, so there aren't those elongation, that kind of striation that we see with cardiac and skeletal muscle. And then there's the presence of the dense bodies, and the dense bodies are somewhat synonymous with the Z disc, and I'll point those out a little bit later on. So structurally, skeletal and cardiac muscle are similar, but functionally, smooth muscle and cardiac muscle are similar as well. So I usually encourage students to draw a Venn diagram when it comes to muscle, just so that you can point out which types of muscles share certain features and which types of muscles don't, right? Really important for you to be able to distinguish which ones have gap junctions, which we'll talk about in a bit, which ones have sarcomeres, etc. They all have actin and myosin, all right? So I'll point that out. They all have actin and myosin. Okay, so the sliding filament mechanism still exists here with smooth muscle, but it's a little bit different. The actin and myosin filaments are longer. In smooth muscle, they're arranged obliquely, so kind of slanted, as we can see here in this drawing, this illustration. And then the myosin heads are longer, so are the actin filaments. And so because these filaments are longer and are arranged in an elongated sort of oblique manner, the range of contraction is also longer. So because the sliding has a long range to occur across, then the time that it takes for this contraction to complete is also going to be longer as well. So longer contractions, longer filaments, which cause those longer contractions. And then we've got these dense bodies. So the dense bodies are basically the areas where the filaments are anchored to each other, but they're also anchored to the connective tissue of the muscle cell itself. And so the dense bodies is what creates the crumpling of this cell when these filaments slide past each other. And so we don't see the shortening that we see as such with the sarcomere, but we see more of a crumpling, this kind of shriveling up of the cell where the contractions are happening really around those dense bodies, those points of attachment between the filaments and the connective tissue of the muscle cell itself. Let's talk a little bit about the steps of smooth muscle contraction. So what happens during excitation, contraction, coupling for this type of muscle. And I'll be pointing out some differences between what happens with smooth muscle, excitation, contraction, coupling, and what happened in skeletal muscle, excitation, contraction, coupling. So the first and the most obvious difference is the source of calcium. In skeletal muscle, we saw that calcium only came from the sarcoplasmic reticulum. Here with smooth muscle, and we'll see this with cardiac muscle as well, calcium comes from two sources. There's extracellular calcium, coming into the cell. There's also calcium in the sarcoplasmic reticulum as well. So there are two sources of calcium for muscle contraction in cardiac and smooth muscle. We've got the same voltage gated calcium channels on the plasma membrane. We've got calcium triggering the release of calcium from the SR and from outside of the cell. So that same calcium-induced calcium release that we saw with skeletal muscle is also occurring here. Calcium binds to calmodulin. Now, calmodulin is a protein that we don't see over in skeletal muscle, and it's really important for smooth muscle contraction. And it forms this calcium calmodulin complex, which then activates another protein or enzyme rather in the cell, which is myosin light chain kinase, or MLCK. When we phosphorylate the myosin heads, this is done by MLCK. And then when we dephosphorylate those myosin heads, those are done by phosphatases. And so MLCK is going to bring about cross-bridge cycling, whereas the opposite enzymes, which are phosphatases, are going to decrease cross-bridge cycling. And this is how we regulate muscle contraction here. So let's kind of go through this slideshow here and kind of point those things out. So we've got two sources of calcium for smooth muscle contraction, calcium coming in from the extracellular environment, calcium also being stored here in the endoplasmic reticulum. And I'm referring to this as endoplasmic, sarcoplasmic. Those are kind of interchangeable here in smooth muscle. So that calcium floods the cytosol itself, or the sarcoplasm. It's then going to bind to calmodulin. This is forming this calcium calmodulin complex, which is this molecule here in purple. The presence of that calcium calmodulin complex is what activates or turns on MLCK, MLCK standing for myosin light chain kinase. And because it's a kinase, we talked about this, kinases add phosphates, phosphatases remove phosphates. So MLCK is going to phosphorylate the myosin heads. Remember that the myosin heads are pretty similar here to what we've seen in skeletal muscle. Having that phosphate on them is what turns them on to drive cross-bridge cycling. And so the act of MLCK phosphorylating the myosin head activates that myosin head, activates cross-bridge cycling, and brings about contraction. On the other hand, we've got phosphatases, so you can write in here, this should be phosphatases. Phosphatases are going to remove the phosphate head from the, the phosphate from the myosin head. And so that is going to result in no myosin ATPase activity, and that is going to cease cross-bridge cycling and cease muscle contraction, okay? So again, the calcium calmodulin complex turning on MLCK, that can either phosphorylate the myosin heads or dephosphorylate them. And if we dephosphorylate, we cease cross-bridge cycling, whereas if we phosphorylate, we bring about cross-bridge cycling and bring about contractions. Now, what I want to point out here is that calcium is also regulating this process, but in a very different way. In skeletal muscle, we saw that calcium regulated muscle contraction by binding to troponin and, uncovering those myosin binding sites on actin by the sliding of tropomyosin away from those binding sites. And here, calcium is also playing a crucial regulatory role, but it's different. It's binding to calmodulin, activating MLCK, and then phosphorylating those heads, or in the absence of calcium, phosphatases can come about because the MLCK is not active, and then we can dephosphorylate those heads and cease muscle contraction that way. All right. And in the interest of time, I'm not going to draw this out, but I highly encourage you to sit down and try to draw this out on your own from your own understanding or memory. And think about the players here. Think about the crucial role of calcium and then what turns the muscle contraction on versus what turns those muscle contractions off. And remember, turning muscle contraction on is MLCK, but then turning it off is basically relaxing of that muscle that happens via the phosphatases. So let's kind of talk a little bit here again about relaxation of smooth muscle. So it's going to require the phosphatases to remove the phosphates from myosin. For this to happen, calcium must leave the cytoplasm, a very similar phenomenon that we saw with skeletal muscle. And this is how calcium is regulating this whole thing. Although it's binding to a different molecule in this cell, it's still the active player that's turning this on and turning it off. So when all that calcium is removed from the cytoplasm, then those phosphatases will remove those phosphates from the myosin heads, and then we can cease muscle contraction. Now, this is done via the calcium ATPase pumps that are pumping calcium back into the endoplasmic reticulum, but there's a second pump here that is also removing the extracellular calcium. So remember, we had two sources of calcium, and so we not only need to put calcium back into the endoplasmic reticulum, but we can also exchange it on the cell membrane for sodium. So there's a calcium-sodium countertransport or calcium-sodium exchanger that basically exchanges calcium for sodium. Remember, sodium wants to get into the cell according to its ECDF, and so we can simply take that energy, right? Remember what countertransport is. It's a secondary active transport mechanism, and so we're simply going to take the energy from sodium trying to get into the cell and use that energy to pump calcium out of the cell. Also here, we want to just point out that the contractions are longer, they're slower. We talked about those longer filaments that give a longer range of sliding. And so the myosin ATPase contraction is also slower here. And so that just makes smooth muscle contractions overall slower, much more sustained, if you will, than what we saw with skeletal muscle contraction.